Hello dear students, myself Dr. Sachin Gurule, Assistant Professor from Department of Zoology, KTHM College, Nasik. So in this video we are dealing with the subject of practical zoology, and within that paper we are going to start with the practicals belongs to the section second, that is animal ecology. And the first practical is the estimation of dissolved oxygen from given water sample by the Winkler's method. So let's see. So first of all, aim of this practical is estimation of dissolved oxygen from given water sample by the Winkler's method. Now as you know that the oxygen is present in the atmosphere into the gaseous form, but in the aquatic water bodies, whatever water is present, that water also contains a oxygen, but that oxygen is in the form of dissolved form. And so that dissolved amount of the oxygen present into the water sample can be measured by the different method, and one of the most standard method is used to determine or estimate the dissolved oxygen in particular water sample. This is given by the Winkler's method. So first of all, let's see the principle behind this experiment. The Winkler method is based on two oxidation and reduction reaction, and this is widely used and is a standard mean of determining the dissolved oxygen from any given water sample. So this method is based on the fact that sodium hydroxide, it reacts with the manganese sulfate to give the white PPT of manganese hydroxide. So here you can see the reaction of this experiment. So in presence of oxygen in highly alkaline solution, the white manganese hydroxide is oxidized to the brown colored manganese oxyhydrate, that is MnOH twice. So this occurs in direct proportion to that of the amount of oxygen present into that particular sample, and that may be judged from the intensity of the brown color PPT which is get formed. So initially the reaction is as follows: the MnSO4 or plus 2NaOH is going to convert into the manganese oxyhydrate. Then it will give first of all the white PPT, that is of the Na2SO4, and then in the further reaction this manganese sulfate in presence of oxygen is going to convert into the manganese oxyhydrate, which gives out a brown colored PPT. Then in that particular brown colored PPT solution in strongly acidic media, the manganic ions are free and they are react with the free iodine ions of the potassium iodide to form a free iodine. Now that when the iodine is liberated, that is going to form a yellow color solution. Now the amount of free iodine is equivalent to the amount of oxygen present in the sample. That means whatever particular amount of the iodine which is get formed in that particular solution after the reaction, it is exactly equivalent to the number of or the dissolved oxygen present in that water sample. So by calculating the amount of the iodine which is liberated, we can easily determine the presence of oxygen in that particular sample. So this amount of the iodine can be determined by the titration with that of the sodium thiosulfate, and the reactions are as follows. So here we have already learned in the previous slide, that is manganese oxyhydrate, if it is given the acidic medium by addition of a strong acid, it is going to convert into the manganese sulfate, and in presence of the particular potassium iodide solution or the azide solution, it is going to form the free iodine, and once the iodine is get formed, that solution is converted into the yellow colored solution, and this liberated iodine is equivalent to the number of amount of oxygen present in that water sample, can be measured by titrating this liberated iodine with that of the sodium thiosulfate, and the after the end point of the titration, with the help of formula we can able to count the iodine which is present into that particular reaction, and ultimately we can able to calculate the amount of oxygen present in that water sample. So this is the principle behind this experiment. So for this experiment, we required the different things. For example, glass stoppered bottle, that is BOD bottle, biological oxygen demand bottle, that is commonly known as a BOD bottles, which are available in two different quantities, 250 ml and 300 ml. So for this experiment, we are using we are going to use the 300 ml BOD bottle. Then you will require the beaker, conical flask, measuring cylinder for measuring the chemicals, then burette, pipette, reagent bottle, and water sample from which you have to estimate the amount of oxygen, dissolved oxygen present in that particular water, and the burette stand. While the chemical reagents you require the Winkler A solution. Winkler A solution is the solution of manganese sulfate, and the second one is alkaline sodium or the potassium iodide solution, which is also referred as a Winkler B solution. Then third chemical you will require sodium thiosulfate, and for this experiment we are using the concentration of sodium thiosulfate is 0.025 normal, and you also require the 1% starch as the indicator solution. So this is about requirements and the chemical reagent. So let's see first of all the procedure. So first of all, you have to take a 300 ml of the pond water in closed BOD oxygen demand water or BOD stoppered water. Now after taking the water sample into the BOD bottle, you have to add the 2 ml of each of Winkler A and Winkler B solution one by one by inserting the pipette just below the water level. You will observe first the white and the brown pipette respectively after the addition of these two reagent, Winkler A and Winkler B. So in the 300 ml of water which is present into the BOD bottle, you have to add 2 ml of each Winkler A and Winkler B solution. Now then after, you have to add the concentrated H2SO4 drop by drop to this content till whatever PPT, brown PPT which is get formed into the first procedure, that it is completely dissolved. You will observe the clear yellowish brown solution indicating liberation of the iodine. So yellow color is representing the liberation of iodine. Then after complete dissolution of that brown PPT, the yellowish brown color is formed in that particular solution. Now from that solution you have to take exactly 50 ml of this yellowish brown solution with the help of measuring cylinder from that stopper bottle into a conical flask. Now into that 50 ml solution you have to add 3 to 4 drops of the starch indicator, and after addition of this indicator you will get a bluish green color which is developed into the solution, or you may observe the yellowish color solution only. So in the next step you have to titrate this solution against the 0.025 normal sodium thiosulfate solution till the solution becomes colorless. Means the end point of this titration is the bluish green or yellowish to the colorless. Now when you are going to titrate with the sodium thiosulfate, once the color of that solution is disappear and color becomes the color of the solution becomes colorless, at that time you have to note down a reading of the consumed volume of the sodium thiosulfate which is used to complete that reaction. And similarly you have to take 2 to 3 readings for minimizing the errors, and repeat this procedure 2 to 3 times and note down your result into the observation table. So before we start with the observation table, so let's see whatever procedure just we have discussed. We are going to see this with the help of one video. So let's see this video first.
So today we will be determining for dissolved oxygen, that is DO. So as you can see here, we have prepared the reagents before as per the procedure given in the manual. The reagents are manganese sulfate, alkali iodide azide, and then starch indicator, and we will be taking standard sodium thiosulfate in the burette, and we have taken concentrated sulfuric acid. So all the reagents are prepared using the procedure given in the lab manual. So as a sample, we will be taking STP water, that is sewage treatment plant water. So this is a BOD bottle of 300 ml capacity. So the sample is being filled in the BOD bottle. It is filled completely. Now the reagents, that is manganese sulfate and alkali azide of 2 ml will be added. 2 ml of manganese sulfate is measured. And is added to the sewage sample. This is alkali iodide azide. 2 ml of alkali iodide azide is measured. This is added to the sample. As you can see, there is air bubbles in that. A tiny air bubbles are created. So before titrating it, mix it well until the air bubbles are vanished and then allow it to precipitate. It is being mixed well in order to eliminate the air bubbles which are trapped inside. Once all the air bubbles are eliminated, allow it to precipitate before the titration. After a while, you can see that it has precipitated completely. Now we'll add concentrated sulfuric acid. So 2 ml of concentrated sulfuric acid is added to the sample after the precipitation. So you can clearly see that it is turned into dark straw color. Now exactly measure 203 ml of sample from the BOD bottle and take it to the conical flask. So exactly 203 ml of sample is taken from the BOD bottle, the conical flask, to titrate it against sodium thiosulfate solution. So now the solution will be titrated against standard sodium thiosulfate solution until it turns to pale straw color. So as you can see, from the dark straw color, it has turned to pale straw color. Few drops of starch is added to the titrated solution now. After the addition of starch, it has turned to dark blue color. Now titrate it until it appears to be colorless. So you can observe that from dark blue color, it has completely appeared to be colorless. Note down the burette reading, that is the volume of standard thiosulfate consumed, in order to find out the amount of dissolved oxygen present in the solution using the formula given in the manual.
Okay, so in this way you can complete the procedure. And coming to the observation table. Now for observation table, so first of all, in the burette, first we are going to take the sodium thiosulfate solution, that is the concentration of 0.025 normal. Then in the second burette you have to water sample, or you can take that water sample directly from this yellow solution, exactly 50 ml of the sample into the conical flask. We are using the starch as an indicator, and the end point is blue or yellowish to the colorless. So this is the observation table. You have to take a 50 ml of the water sample from that into the conical flask from that BOD bottle which we have already proceeded, and you have to titrate the solution against the sodium thiosulfate, and you have to take at least 3 readings, and after taking the 3 readings you have to calculate the mean burette reading for the calculation. So the calculation is very simple. So 1000 ml of one normal sodium thiosulfate is equivalent to the 8000 milligrams of the oxygen. So volume of the thiosulfate, that is 0.025 normal, is equal to V into 0.025 into 8000 divided by the 1000, which is for the milligram of oxygen present per 50 ml of that water sample. Now dissolved oxygen can be calculated by the formula by mean burette reading into the concentration of the sodium thiosulfate we are used into 8 milligrams of oxygen into 1000, because whatever result we have to write, we have to express it in per liter. So here we are multiplying it by the 1000. So this divided by the 50 ml of sample. So why 50? The ml of sample which is used for the purpose of titration. So you can minimize the formula as like this one. The dissolved oxygen is equal to V in the sense the volume of the sodium thiosulfate used, which is obtained from the mean burette reading, into 0.025 into 8 into 20 milligrams of oxygen. So by this formula you can able to calculate the amount of dissolved oxygen present in your water sample. So let's see one example. So for example, by the procedure just we have discussed, we obtain the readings such as 2.2, the first reading, second reading is 2.1, and third reading is also 2.1. So from these 3 readings we have to calculate the mean burette reading, and that can be calculated by addition of these 3 readings and divided by 3. So mean burette reading for this sample is 2.133. So this figure we have to put into the formula just we have discussed. So dissolved oxygen, that is 0.025 normal, is equal to V into 0.025 into 8 into 20 milligrams of oxygen. So by this formula you can able to calculate the amount of dissolved oxygen present in your water sample. So you can minimize the formula as like this one. The dissolved oxygen is equal to V into 0.025 into 8 into 20 milligrams of oxygen. So this figure we have to put into the formula just we have discussed. So dissolved oxygen is equal to 2.133, which is a mean buried reading, into 0.025 normal, which is the concentration of the thiosulfate we have used, into 8 and into 20 milligram of oxygen. So by doing such kind of the multiplication, you will get an answer of 8.532 milligrams per liter. So with the help of this method, you can easily calculate the amount of oxygen dissolved in a water sample from any water body. So the result of this experiment is amount of dissolved oxygen present in given water sample is 8.532 ml of oxygen per liter at normal temperature and pressure. So in this way, today we have learned in this video how to estimate the dissolved oxygen from the any water sample by using a Winkler's method. So in your journal, you have to write the aim, then requirement for this experiment, principle, observation table, and calculation, and lastly you have to write the result like in this written in this slide. So thank you very much.